Greetings children, welcome to the classes. Today we are going to learn the last part of photosynthesis, and today's class I'm going to show you all the experiments that you need to learn as per syllabus on photosynthesis. There are various experiments, but still you need to go with 5 different experiments. As per your syllabus you are learning just 5 different experiments. Okay, so the first experiment is starch test. Starch test means it shows that how we are going to test the presence of starch in the leaf. Okay, the presence of starch in the leaf. So this plant is coleus plant, the same plant which is mentioned in your lesson, coleus plant. And this plant has been selected and they have detached just one leaf, and what they are going to do is, before doing this starch test, what they need to do is they need to de-starch the plant. They have to keep that entire plant in dark. Generally they are preferring to choose potted plant. The potted plant means they can move it as they wish. So from a small potted plant they will be selecting a small potted plant, and this potted plant will be moved into dark for 24 to 48 hours. That means from 1 day to 2 days we need to keep this in dark. The reason for this making this plant in dark or placing this plant in dark is the entire starch found in this plant will be used up. There is no photosynthesis for those 2 days. So for the survival, the plant will be using the available starch in the plant. So the plant, we are making that plant as starch free plant. Okay, the plant is going to be starch free. That's what the plant is going to be, starch free. Now what she is doing is she has plucked a leaf from the de-starched plant, and this large beaker, there is a large beaker and there is a small beaker too, and in this large beaker she is going to do the test. This is the small beaker. She is just showing you the small beaker now. Now she is going to take hot water, boiling water. She is going to boil it again. So this is the first step of starch test. Dip the leaf in boiling water for a minute, minimum for a minute. The purpose behind this is we are going to kill the cells. This is very important. You are placing the plant in boiling water to kill the cells. Okay, to kill the cells.
Have enough water can indeed, in the presence of carbon dioxide, and they are autotrophic and photosynthesized. So what we will be testing to see is whether green plants that contain chlorophyll are in the presence of carbon dioxide, have enough water, can indeed produce their own food. What I need to do is I need to gather all apparatus. I need a large beaker. If you don't have a large beaker in your lab, you could actually use a jam jar, a large jam jar. I also need a smaller beaker in which I'll be placing ethanol. So if you don't have one of those, you could substitute it with a fish paste jar. Now to start off, I'm going to be pouring boiling water into this beaker, and I'll place it onto the hot plate. Many labs have Bunsen burners, so you could be using that to heat the water. And you place the leaf into the boiling water. Now the reason why we do that is we want to stop all metabolic processes, and we want to soften the walls, the cell walls of the plant. Now we'll leave it in that boiling water for a minute or two.
Now the purpose, you need to make sure that the purpose of keeping or dipping the leaf in boiling water is to kill the cells. Okay, this is to soften the cells, yes, of course, but it is the main purpose is to kill the cells so that later the chemicals can enter into the cell. Okay, the main purpose is to kill the cells. First, we are dipping the leaf in boiling water to kill the cells.
And allow it to soften those cell walls. Now, the reason why I place a test tube on top of it is just if I were to leave the leaf, it will float upwards, and just to make sure that it's getting maximum heat from the plate. What I'm going to do now is I'm going to remove the leaf with a tweezer, take it out of the boiling water, and place it into the smaller beaker. And now I need to use some ethanol. Ethanol will extract the chlorophyll out of the leaf. Because when we want to test for starch, we need the leaf to be pale. Now we need to heat the ethanol. If you are using a Bunsen burner, be sure that before even touching the alcohol, you have switched off the burner, as alcohol is highly flammable. Now I place the beaker with the ethanol, which has the leaf in it, into the hot water, because ethanol needs to be heated slightly. And I gently, with the back of a test tube, just tap the leaf gently to help with the extraction. You could just leave the leaf in the ethanol, which is heated slightly by the hot water, but I know that if we tap it gently, this process is just a little bit quicker.
Now the purpose of this use of ethanol or methylated spirits, both are similar. Ethanol or methylated spirit is same only. Methylated spirit is also ethanol, which is mixed with some amount of methanol or butanol or some other substances to make it smell bad, to make it non-consumable. That is the purpose. So ethanol or methylated spirit are same only. So she is using ethanol here. In your text it's given methylated spirit. And why we are using methylated spirit is to make it pale. Okay, the purpose of using methylated spirit is to make the leaf pale. That means we are going to take all the chlorophyll out of the leaf. We are going to remove the chlorophyll from the leaf. By the end of this test, the leaf becomes hard and brittle. By the end of this treatment of methanol, the leaf becomes hard and brittle.
Careful not to tear the leaf. So it must just be a gentle tap. Now what you will see happening is that the ethanol is turning a light green, and when we take this leaf out, we should see that the chlorophyll has been extracted, and we will have a pale leaf to see the color change much easier. Now when I lift this out, you will see that the ethanol has turned green, and the leaf is a pale color.
Can you check? The leaf is so strong, rigid.
I now need to rinse the ethanol off. So I'll place some water onto it. Before I can add the iodine solution,
so the third step of this experiment is place the leaf, while you take it out from methylated spirit, place the leaf again in water. The reason is to bring it back, to make it soften. Okay, so first what we have done, or what she has done, is she placed the leaf in boiling water for a minute to kill the cells, and then she placed the leaf in methylated spirit or ethanol to remove the chlorophyll content from the leaf so that the leaf become pale. And then third thing, she placed it again in hot water to make it soften. Okay, again she pour water onto the leaf to make it soften.
You need to rinse this water off. Pour it off petri dish. Now when testing for the presence of starch, we use iodine solution. Iodine solution in the presence of starch will turn to blue-black.
So here iodine is the indicator of starch. Iodine is the indicator of starch. Okay, starch, name the starch indicator. Sometimes people used to ask question like that. That is, iodine is the indicator of starch.
Place a few droplets of iodine solution onto the discolored leaf. And I can see that the brownish color has turned darker to a blue-black color, indicating that starch is indeed present in this leaf. So we know for sure that photosynthesis has occurred.
Now by this experiment, what we have understood is, while you pour iodine solution, the indicator, over the leaf after all the treatment, if the leaf turn into blue-black color, it indicates the presence of starch. So only chlorophyll, if it is present, starch should be produced. If chlorophyll is not there, starch will not be produced. Okay, so this is starch test. This doesn't shows that this is because of chlorophyll, or this is because of carbon dioxide, or this is because of sunlight. This is just a starch test. How to do the basic starch test. Okay, we are going to learn all the experiments in future. Again we are going to learn the experiments. In all these experiments, finally we need to go with starch test. So this is the procedure for starch test. Okay, so this is the basic for all the starch test. After your experiment, specific experiment, we have to go with starch test. We are detaching the leaf, we are boiling it in hot water to kill the cells. It's very sure, you have to write it as kill the cells. Some people, like when we are hearing about starch, like when we are hearing about hot water treatment, people used to think of they will be writing as to kill the germs. Okay, generally people used to write like that. Why we are placing the leaf in hot water, they will be writing it as to kill the germs, but that's wrong. You should mention it as kill the cells. Okay, so that is first step. Place it in hot water, dip it in hot water for 1 to 2 minutes to kill the cell. And second step is place it in methylated spirits, and that is to remove the chlorophyll. And third step is place it again in boiling water, that is to soften the leaf or soften the cells, and then pour iodine on top of it. If the leaf is changing into blue-black, that is starch is present. If the leaf is in brown color, there is no starch. Okay, so these are the 5 major points for starch test. Shall we go with the first experiment, that is chlorophyll is necessary for photosynthesis. Now this is a coleus plant. Okay,
pluck a coleus leaf from plant. Coleus leaf contains both green and non-green areas. Transfer.
Why they have selected coleus leaf, or you can go with even croton leaf. This coleus or croton leaf will be showing variations, like in a leaf completely it won't be in green color. There you can see brown color, there you can see non-green parts also. That's why they have preferred this coleus leaf. So sometimes in questions they will be asking like variegated leaf. Variegated leaf means the leaf which doesn't show only green part. Okay, that is what coleus leaf, why we are going specifically with coleus leaf.
The leaf on a white sheet of paper. and draw its outline. Mark the green and non-green areas carefully on it. Now boil this leaf in water to kill it. After boiling in water, again boil it in alcohol.
Can you see that? This
The
is just
purpose
starch
of boiling
test.
leaf in alcohol is that it dissolves greenish part, that is chlorophyll. Then wash the leaf in cold water and place it in a watch glass. Now pour some drops of iodine solution over it. Compare the leaf with the drawing you had made. Observation: only those areas of the leaf become blue which were marked as green. Conclusion: this shows that chlorophyll is necessary for photosynthesis.
Now the third experiment. Now we have discussed two different things. One is starch test, and then the other one is chlorophyll is necessary for photosynthesis. The next experiment is experiment to prove light is necessary for photosynthesis.
Take a potted plant with wide leaves. Keep it. In these weekdays, photosynthesis does not take place in the plant due to unavailability of sunlight. Hence, the starch which is present in leaves consumed by the plant. Now, leaves are free from starch. Then select a leaf and cover half of the leaf with a piece of black paper by using a clip. Keep the entire setup in sunlight for 4 to 5 hours. Here, the part of the leaf does not expose to the sunlight, which is covered by the black chart. Detach the leaf from plant. Remove the clip and chart from the leaf. Boil the leaf in methylated spirit over a water bath till it becomes pale white. Then test the leaf by using iodine solution. Part of the leaf which is exposed to sunlight turns into blue-black color. It indicates that photosynthesis takes place in this part. Part of the leaf which does not expose to sunlight doesn't turn into blue-black color. It indicates that photosynthesis does not take place in this part. So, this experiment proves that light is essential for photosynthesis. Okay.
Yeah. Thus, we have seen the experiment which shows sunlight is necessary for photosynthesis, and the next experiment is carbon dioxide is necessary for photosynthesis. Okay, CO2 or carbon dioxide is necessary for photosynthesis.
Keep it in a dark place for weekdays.
For destarching.
In these weekdays, photosynthesis does not take place in the plant due to unavailability of sunlight. Hence, the starch which is present in the leaves consumed by the plant. Now, leaves are free from starch. Take a glass bottle with 5 to 6 ml of potassium hydroxide solution. Insert half of the leaf into the glass bottle by using split cork. Close the bottle without leaving any gap by using grease. Hence, air does not enter into the bottle. In this arrangement, carbon dioxide is available for the part of the leaf which is present outside the bottle, while carbon dioxide does not available for the part of the leaf which is present inside the bottle. Why? Because potassium hydroxide solution absorbed carbon dioxide present in the bottle. Keep the entire setup in sunlight for 4 to 5 hours. Hence, photosynthesis starts in the leaves of the plant. Detach the leaf from plant. Carefully remove the leaf from bottle. Over a water bath till it becomes pale white. Part of the leaf which is present outside of the bottle turns into blue-black color. It indicates starch is produced in this part. That means photosynthesis occurs in this part. Part of the leaf which is present inside the bottle doesn't turn into blue-black color. It indicates starch does not produced in this part. That means photosynthesis does not occur. So, this experiment proves that carbon dioxide is essential for photosynthesis.
Okay, by this experiment we have seen carbon dioxide is necessary for photosynthesis. So here what are the things you need to note down is KOH is used as an absorbent of carbon dioxide. Instead of KOH, you can be suggested to use lithium hydroxide or sodium hydroxide, any of the hydroxides. So any of these hydroxides can be used as a chemical to absorb carbon dioxide. And the next experiment what we are going to see is oxygen is evolved during photosynthesis. Oxygen is evolved during photosynthesis. This is hydrilla experiment. We are going to use hydrilla plant for this experiment. Let me play this video for you.
The test tube and funnel experiment. The test tube funnel experiment demonstrates that oxygen is evolved during photosynthesis. A few branches of hydrilla are kept in a beaker containing pond water in which a small amount of sodium bicarbonate is dissolved. The branches are covered with a glass funnel.
The reason for using sodium bicarbonate is to give more amount of carbon dioxide. Okay, sodium bicarbonate is used in photosynthesis experiment to release more carbon dioxide.
And a test tube full of water is kept inverted over the stem of the funnel as shown in the diagram. Now the apparatus is kept in sunlight for 4 to 6 hours. The gas bubbles may be observed from the ends of hydrilla branches kept within the glass funnel. These gas bubbles are collected in the test tube by the downward displacement of water. The gas is tested for oxygen. When a burnt splinter is taken near the mouth of the tube, it glows brightly and proves that the gas is oxygen. The test tube and funnel experiment demonstrates that oxygen is evolved during photosynthesis.
So this is the last experiment. So totally we have seen 5 different experiments. The first one is starch test. This is the basic for all the tests, and then chlorophyll is necessary for photosynthesis, in which we use Coleus plant, and light is necessary for photosynthesis, which is a black paper test, and then carbon dioxide is necessary for photosynthesis, that is by using KOH, which is in the bottle, by using a split cork, that experiment, and then oxygen is produced during photosynthesis, that was by using hydrilla plant. So these are the 5 different experiments you need to learn in this lesson. From this experiment, it's possible to ask part B questions, which is having 5 marks together. So please read, go through the entire thing as it is given in your book, and mark down or note down the points what I have said to you. So this is your homework. You have to read the entire text, and this week cycle test on Thursday you will be writing questions from this lesson. This is for 10B. If 10A is also allowed this homework, you can write. So 10B, this is your portion for cycle test on this Thursday. So that's it about this lesson. So we have completed photosynthesis lesson, and from next class onwards we can move on to the next lesson. If there is any doubt, message me back in the personal comment or else in my WhatsApp number. Thank you, children.